Hello, and welcome to the DNA of Onion Lab for Chemistry 102. First, I'll do a quick overview, give you a look at all the stuff we're going to use. First, we're just going to start simply with cutting up an onion, dicing it into little bits, and weighing out 50 grams for this experiment. After we get that 50 grams in a beaker, we add some homogenizing solution, and then we incubate it at 60 degrees for 15 minutes. Once that 15 minutes is done, we quickly transfer it over to an ice bath, and we want it to cool to between 15 and 20 degrees. So we check it with a thermometer. After it's cool enough, we're going to transfer it into a blender, and we're going to do a little low speed, 45 seconds and 30 seconds at high speed. Once we're finished blending, we're going to pour the foamy mixture into a larger beaker and then put that back in the ice bath for another 15 to 20 minutes. We're trying to get the foam to settle and keep it cold. Now we rigged up a little filter in another beaker with 4 layers of cheesecloth, and we'll pour the foamy mixture over the top of this, allowing the liquid to get into the original beaker. Once we double check that the filtrate is still 10 to 15 degrees with our thermometer, we're going to try to precipitate the DNA by adding ice cold ethanol. So we're going to form a layer of ethanol on top of the water mixture. Doing this, the DNA is the only thing not soluble in ice cold ethanol. Everything else should stay in solution. Using a little glass J-hook that we made from a pipette, we're trying to grab out little pieces of DNA, which are white stringy looking things, and as soon as we get enough of those out, we can move on to the next part of the experiment. The DNA that we isolate, it looks just like a white, kind of blob looking stuff. And we're going to then compare that to the protein from an egg white. So in our next series of tests, we're going to take 3 test tubes with DNA in it and 3 test tubes with egg white, and we're going to compare what happens to each. In the first test, we just add a little water to each tube and we put it in a boiling hot water bath. You can see the egg white on the right side starts to cook and get solid and white. Meanwhile, the DNA seems to be floating and dissolving. In our second experiment, we're doing the acid test. And this just means we're adding some hydrochloric acid and then shaking the test tubes. In our final test, we're doing the Biuret reagent and shaking those tubes. So our final results with the temperature test are shown here. With the acid test, the DNA seems pretty much all dissolved, and the egg white just turned into a solid white mass. On the Biuret test, one of them, the protein side, turned purple, and the DNA had no color change. Here's a final look at all of the test tubes. which you can take and use this information to answer any of the questions on your DAS. As before, if you need help, check in with your online TAs. Thank you for watching.